So what do you mean by principles of inheritance and variation? That is, you would have heard about the many things about genetics, right? So what is genetics? That is, it is about the genes and chromosome. That is how the characters are transferred from the parent to progeny, right? So in this chapter, we'll study about the important terms which are related to inheritance and variation. And also we'll study about the monohybrid cross. So in this class, we'll discuss about the important terms and about the monohybrid cross and about the Mendel's law of inheritance, and under that the main laws are law of dominance and law of segregation, right? So these are the main topics that we are going to discuss today. Okay, so first, what is genetics? So genetics is actually, it is a study of inheritance. That is how the characters are transferred from parent to progeny, right? And then heredity and variation of characters. Inheritance, that is heredity and variation of characters, or study of genes and chromosomes. So it is called the genetics. And you would have heard of the scientist, that is Mendel, Richard John Mendel. He is the father of genetics, right? He is the father of genetics. And actually the term genetics was coined by the scientist, that is Batson, William Batson. He coined the term genetics, and he is called the father of modern genetics. Of modern genetics. Okay, so William Batson, he coined the term genetics, and he's called the father of modern genetics. And there are several other scientists like Morgan, and he's called the father of experimental genetics. That is, he did a lot of work on linkage and crossing over. Morgan's, he did work on Drosophila, right? So Mendel is the father of genetics. So it is the study of inheritance and the variation of characters. Okay, so what do you mean by inheritance? That is, it is the transmission of characters from parents to progeny. So how the characters are transmitted from parents to progeny, and it is the basis of heredity. Okay, so this genetics actually deals with the variation, that is, and to what degree there occurs a difference between the parents and the offspring. That is the variation, right? Difference between the parents and the offspring. So for example, if we take a particular character, that is eye color, right? So that is a character. It is a heritable feature among parents and the offspring. That is, you may be heard of the fact that you are looking like a father or looking like a mother. So how it is happening? It is due to the transmission of that particular character through the genes, right? So it is a heritable feature. That is, it is transferred from parent to progeny. It is among the parents and the offspring. For example, eye color is a character. So what do you mean by a trait? It is a variance of a character. For example, if we take a particular character, that is eye color, it may be a brown eye, blue eye, or a black eye. So these are the different traits, right? So they are the variance of a particular character. It is called a trait. So these are the important terms. That is, what is genetics, what is inheritance, and what do you mean by variation, what are characters, and what is a trait, right? So these are the important terms. So all the progeny, that is in the case of every organism, right? So if you put a mango seed, it gives rise to a mango plant, and elephant, it gives rise to a baby elephant. And even in human beings, you are resembling either of the parent, right? So all these are due to the inheritance of the genes from parent to progeny, right? So even in olden days, that is, the people were aware of the inheritance of genes, right? So for example, they produced a variety, that is a well-known Indian breed of cow, that is called the Sahiwal cattle of cows in Punjab. So how they made this breed. That is, it is a well-known Indian breed, and it is actually produced through the artificial selection and also domestication from the ancestral wild cows. So from the wild cow, they analyzed the best features and they selected those features and artificially selected and domesticated, and they produced the best breed, that is Sahiwal cow. So even in olden days, people were aware that that variation, it happens through the sexual reproduction, right? So analyzed all the wild plants and animals, and they were able to make out the best breeds, though they are not aware of the scientific basis behind this inheritance, right? But they were aware that the variation occurs through sexual reproduction. Okay, so Mendel is the first scientist. Actually, he is the father of genetics, and he proposed the various laws, right? So they are called Mendel's law of inheritance. And actually, his work was not recognized until his death. He published all his work in a journal, that is a German journal, but it was not recognized in his period because of the lack of communication, and also he used many of the mathematical approach, right? He used the many mathematical calculations in biology, and that was not accepted in those days. But only the later scientists, they rediscovered these Mendel's laws, and they proposed the law of dominance, and then law of segregation, and law of independent assignment, right? And what Mendel did, he chose the garden pea plant. So why he chose the garden pea plant? Because those pea plant, actually, they were able to grow in a shorter period of time. That is, the first main feature is the garden pea plant. It's an annual plant, right? It is a annual plant, and it is a lifespan of about 2 to 3 months. So large number of offsprings were produced. Within the short period of time, he was able to analyze the large number of plants. So that is one of the advantage. And the another, that is why Mendel chose the garden pea plants, right? So the main reason is it is an annual plant. Within 2 to 3 months, large number of offsprings were produced. And it has the many contrasting traits, right? It has many contrasting traits. And it is able to self-pollinate, that is natural self-pollination is present. Self-pollination is present, and also hybridization is possible. So hybridization is possible, and it is easy to cultivate. And they have the large seeds. So these are the main properties that the pea plants have. So Mendel selected the pea plants for his experiment, and he did many of the hybridization experiments, right? So he chose the garden pea plant for his studies. So he conducted some hybridization experiments on garden peas, that is Pisum sativum. For 7 years, he did work on the garden pea plant. Okay, so how he made a cross in the pea plant? First, he selected the pea plant, that is selection of pea plants with contrasting characters. So what do you mean by contrasting characters? So if you take the height, tall and short or dwarf, right? So these two are contrasting characters. So he selected the pea plant with contrasting characters, and then emasculation. So what do you mean by emasculation? That is the removal of anthers of one plant to avoid self-pollination. So for example, if it is a bisexual flower, right? He removed the anthers. For example, in this purple flower, he removed the anther alone, right? So he removed the anther of one plant to avoid self-pollination. So self-pollination is avoided. So now it is a female plant because it has only the female reproductive part. So then what he did, that is, he transferred the pollen from anther of white flower to the stigma of the purple flower. So from the white flower, he took only the pollen, right? These pollens are transferred to the stigma of the purple flower. So now he can crossbreed these two, right? The purple and the white one, because here the stigma is from the purple and the pollen is from the white flower, right? So removal of anther of one plant to avoid self-pollination is called emasculation. So this is the female plant, and then he collected the pollen grain from the male parent and transferred to the female parent, right? So after transferring, then what will happen? Cross-pollination now. So the flower produces seeds, right? Now if you plant these seeds, it leads to the production of offspring, and this is called the first generation offspring, right? So he selected the pea plant with contrasting character, then removed the anthers of one plant to avoid self-pollination, and then collection of pollen grain from the male parent and transfer it to the female parent, then collection and germination of seeds to produce offspring. Okay. So actually he selected the 7 true breeding varieties. That is the true varieties. It is a pure line, right? For example, if it is it is tall for so many generations. So similar type of plants he selected. So he selected the 7 characters. So what are the characters? One is the stem height, and then second is the flower color, flower position, pot shape, pot color, seed shape, and then seed color. And if you take the dominant character in the stem height, it is tall, and recessive is dwarf, right? So if you take the stem height, tall, and the dwarf. So based on that, similarly, flower color, violet is the dominant character. If you take the flower color, so this is the dominant trait, and this is the recessive white. And flower position, so based on the position of the flower, if it is present in the axial position, so this is the axial position, that is a dominant character, and the recessive is the flowers at the terminal, right? It is present at the end, so this is a recessive character. Then part shape, that is inflated and constricted, that is the shape of the part. This is full and this is the constricted part. So this is a recessive character. Then pot color, green and yellow. Seed shape, round and wrinkled. So round is a dominant character and wrinkled is the recessive character. And if you take the seed color, yellow and green, right? So you have to be clear with this table, because many questions they may ask from this, that is regarding the character of the flower. So how many characters the Mendel took into account? So regarding flower, 2 characters, right? Color and the position. Similarly, regarding pod, it is shape and color. Regarding seed, shape and color. And also they may ask, so how many colors he took into account? So flower color, pot color and seed color. So you have to be thorough with this table with the details of which is the dominant character and which is the recessive character, right? So many questions may come in this. So you have to be clear with it. So 7 true breeding varieties were selected by Mendel. So they are contrasting traits. So they are of the opposite. One is tall, the other is dwarf. And if it is green, and that is yellow, right? So what is a monohybrid cross? Monohybrid cross. So it is a cross involving 2 plants. So 2 plants are involved. They differ in 1 character pair, because he didn't take many characters into account. So if he is analyzing the height, that is, he took into account only those tall plants and the dwarf plants, right? So it is a cross involving 2 plants differing in 1 character. And then next he analyzed the flower color. So he took into account only the flower color. So he only 1 character is analyzed. So it is called the monohybrid cross. So cross involving 2 plants differing in 1 character. Okay. So for example, Mendel crossed the tall and the dwarf pea plants to study the inheritance of 1 gene. So study only about the height, right? So this is the female parent and removal of anther, and from the male parent, the pollens are transferred to the female. right? So by dusting, so simply by dusting the pollen grains, this will land on the stigma, right? So he studied about the single character first. So he analyzed the actually this or the parental, right? So in the case of pea plant, first he analyzed the two characters, that is two traits, tall and the dwarf height, right? So he took the true breeding pea lines. So what do you mean by true breeding pea lines? That is true breeding line is a one that is undergone the continuous self-pollination. So has undergone the continuous self-pollination. So what do you mean by continuous self-pollination? That is, if you want to choose the tall plant, he wanted it to be of true breeding line. So that is, this plant is tall for so many generations. So it is a pure tall plant. So he took the pure tall plant and a pure dwarf plant, that it was a dwarf plant for so many generations. That is continuous self-pollination. So he took true breeding lines and he crossed the tall parent with the dwarf parent, and actually what he observed is the production of a tall plant, right? When he crossed the tall with the dwarf in the F1 generation, it is also called as first filial generation, right? He got the tall plants in the F1 generation. And when this tall plant is self-pollinated, so it is again crossed with the same plant, same tall plant. And in the F1, that is F2 generation, what he observed? He observed the 3 tall plants and 1 dwarf plant, right? So what does this indicate? That is, in the F1 generation, all the plants were tall, right? But in the F2 generation, he observed that it is in F1, all are tall. But in the F2 generation, he observed the 3 tall and 1 dwarf, right? But none of them were of intermediate height. This tall is equivalent to the parental tall, and here also it is equivalent to the parental, but none of them were of no intermediate height, right? Of intermediate height. So what did he observe from this? That is, these characters are passed from parent to progeny by some factors. That is, some factors are passed from parent to progeny, and these factors are later on called as genes. He referred to it as factors, and later they are called genes, right? So he took the true breeding pea lines, that is, which has undergone the continuous self-pollination, and in the F1 generation produced full tall plants, all the plants are tall. But in the F2 generation, that is the second filial generation, I observed that 3 tall and 1 dwarf plant, but no plants were of intermediate height. So he observed that some factors, or he proposed that something is passed from parent to progeny, and he called it as factors, and now they are called as genes, right? So the next is, so these were actually drawn in the form of a square that is called the Punnett square. So what is this Punnett square? Actually, it is a geometrical representation, or it is a graphical representation. Punnett square is the graphical representation to calculate the probability of, is the graphical representation to calculate the probability of all possible genotypes. All possible genotypes of offsprings in a genetic cross. So it is a graphical representation to calculate the probability of all possible genotypes of offspring in a genetic cross, and it was proposed by the scientist, that is Reginald Punnett. So he proposed the graphical representation. That is the production of gametes by the parents. So if we cross the tall plant and the dwarf plant, so this is the, actually it is represented in the form of a alphabetical letters, right? If to represent the tall, it is represented as T, because it is a dominant character and the recessive character. So it is represented as small t for dwarf. So T, the capital letter represents the tall, and the small letter, it represents the dwarf, right? So if it is a tall parent, it is represented as TT. So this is called the homozygous condition. That is, both are dominant, and this is also homozygous, but both are dwarf, right? So when a tall parent is crossed with the dwarf parent, that is, the gametes are represented as T and T. So T and T for the tall parent, and for dwarf parent it is T and T, right? So this, when they were breed together, when this tall parent and the, that is the gametes of this tall parent and the gametes of the dwarf parent are breed together. So what will be the possible combination? T and this T, so may fuse together and form TT, right? And here this may form again T and T. So here this also will produce T and T. So all are the hybrid, that is, it is called heterozygous. That is, if the one allele is one, this is from one parent, right? And this is from the another parent. So they fuse together. So now if they form a different type of allele, that is different combination, then this is called the heterozygous. So this is homozygous tall, and this is homozygous dwarf, and this is heterozygous tall actually. So only if both are present, the dominant character exists. So it is a heterozygous tall, right? So clear? So this is called the Punnett square. So it is a graphical representation to calculate the probability of all possible genotypes, of offspring in a genetic cross. So this is the genetic cross between TT, that is tall, and the dwarf, right? So whatever is produced here will be of heterozygous tall in the F1 generation. So this is what is represented here. That is, these are the gametes. So they are represented in the Punnett diagram. It is represented in the top and in the side. That the gametes of each parent are represented. So TT, TT. So when they are breed together, the F1 generation all will be heterozygous tall. That is TT, 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 and TT, right? So when this heterozygous tall is crossed with the another heterozygous tall, that is self-pollination, then the ratio that is obtained will be, so when it is crossed, that is T, T and TT, right? So it will be, this will produce homozygous tall, and when this will produce with this heterozygous tall. So this and this will produce again heterozygous tall, and these two T, they will produce the homozygous dwarf, right? So the ratio, that is the phenotypic ratio, is 3 tall. 3 will be tall. That is, though it exists in the heterozygous condition, they are tall. So 3 tall is to 1 dwarf. So this is a dwarf plant. But if you take the genotypic ratio, that is the presence of alleles, whether it is present in the homozygous or heterozygous condition. So TT is 1, and this heterozygous is 2, and TT is 1. So 1 is to 2 is to 1. Clear? So that is the genotypic ratio. So this can also be produced mathematically. That is, without doing this experiment, we can also do it mathematically. That is, gametes bearing the genes T or T in equal frequency, if they are present in the equal frequency of half. So this can be expanded in this form. That is half T plus half the small t to the square. And if you expand this equation, we'll get 1 by 4 of tt plus 1 by 2 of tt plus 1 by 4 of tt. So you can also calculate by means of this mathematical probability, right? So this is what we saw, that is homozygous tall when it is crossed with homozygous dwarf. These are the two gametes that are produced, and if the F1 generation all will be tall, and if it is self-pollinated, they produce the two different types of gametes. That is, this is the Punnett diagram for this cross, and it produces the phenotypic ratio of 3:1, or the genotypic ratio is 1:2:1, right? But this mathematical expression, it cannot actually say about the, that is whether it is a homozygous tall or the heterozygous tall. We are saying that the tall parent, right? That is, it may be either TT or it may be either TT. But mathematical calculations, we cannot predict whether it is a homozygous tall or the homozygous or heterozygous tall, right? So here in the F1 generation TT, when it is self-pollinated, they produce gametes T and T in equal proportion. That is, when TT is crossed with TT, the gametes that are produced are, here it is T and T, and here it is T and T. So equal proportion of T and T are produced. And during fertilization, the pollen grains of T, that is, if this is from a pollen, right? This T have a 50% chance to pollinate eggs of T. So it has the, if this is from egg, right? When the fusion happens, this T has the 50% chance to pollinate either this T, or it has a 50% chance to pollinate this T, right? So 1/4, also the pollen grains of similarly for this T, they have the 50% chance to pollinate eggs of this T and also of this small t. So 1/4 of the random fertilization leads to TT, and 1/2 of the random fertilization leads to heterozygous type, and 1/4 of the random fertilization leads to homozygous dwarf, right? So this is the ratio of monohybrid cross. So now we have to be clear with these terms. That is, what are alleles? They are the alternative forms of a gene. That is, for example, if you take the character of the height, T is the tall. That is, the capital T is tall and small t is dwarf. So these two are called the alleles of the gene. That is T and the small t, they are called the two alleles, right? So they are the alternative forms of a gene. They are called alleles. And what do you mean by homozygous? The condition in which chromosome pair carries similar alleles. That is, if both are same type, then it is called homozygous. So this is homozygous tall and this is homozygous dwarf, and this is for yellow, that is homozygous, and here it is the recessive character, right? And for heterozygous, so what is called heterozygous? The condition in which the chromosome pair carries dissimilar alleles. That is T, T. One will be of the one type, the other one will be of the other type. That is alternative form of a gene, right? So then that is called the heterozygous form. And what do you mean by a dominant character? The character which is expressed in heterozygous condition. So heterozygous condition is, if you take the stem height TT, this is the heterozygous. So the what character is expressed in the heterozygous condition is called the dominant character. So it is indicated by a capital letter, right? So here tall is the dominant character, and that is only expressed when it is present in the form of TT. But this T dwarf or the small letter, it is expressed only in the homozygous recessive condition. But when it is in the heterozygous form, it will not be expressed, right? So the dominant character is the character which is expressed in the heterozygous condition. And what is the recessive character? The character which is suppressed in heterozygous condition, and it is represented by a small letter. So in this case, if you take TT, the dwarf character is suppressed. So this is the recessive character. Only the tall character is expressed, right? So the character which is suppressed in the heterozygous condition is called the recessive character. And then what do you mean by phenotype and the genotype? So visible characters of an individual or the physical expression is called the phenotype. For example, if we observe the plant, we can see that there is 3 plants are tall. By observing, we can find that they are tall plants. And by observing the growth of it, we can say that it is a dwarf plant. So it is a visible character that is called the phenotype. And genotype is the genetic constitution of the character. For example, if visually the plant may be tall, so phenotypically it is tall, but genetically it may not be TT, it may even be T, that is TT, heterozygous form, right? So genotype is the genetic constitution, okay? And what do you mean by hybrid? Hybrid is an individual which is produced by the mating of genetically unlike parents. So if they mate and they produce a hybrid, so sometimes the offspring that is produced will be of more higher capacity than the parents. So that is called hybrid vigor. That is, there will be more puff than the parents, right? So individual produced by the mating of genetically unlike parents is called the hybrid. So in the case of monohybrid cross, the phenotypic ratio is tall is to dwarf, that is 3:1. And if you take the genotypic ratio, it is 1:2:1. Okay. So Mendel actually, he self-pollinated that. We saw up to F2, right? So first he took 2 pure lines and then they form a heterozygous, and if it is this is the F1 generation, and he self-pollinated these 2, that is the heterozygous, and we got the different ratio in the F2 generation, right? So he self-pollinated F2 and found that dwarf F2 plants continue to generate dwarf plants in F3 and F4. So again, he tried with the 3rd and the 4th generation, and he found the production of more dwarf plants, right? So he concluded that the genotype of dwarfs was homozygous. So the dwarfs were expressed only in the homozygous condition, right? That is small p, small p. And apart from studying these height, he also observed on the other traits like flower color, the part color. So all the other contrasting traits also he studied, and he found that some factors were inherited from parent to offspring. So when he studied the different generations, he found that some of the factors are inherited from parent to the offspring, and these factors are now called as genes, right? So what he observed as factors is now called genes. So what do you mean by back cross and the test cross? That is, we saw that as a result of the mathematical explanations, we cannot find that whether the plant is a homozygous tall or a heterozygous tall. So that can be found out by means of a test cross. So what is a test cross? It is the cross of an organism or crossing of an organism with dominant phenotype to a recessive individual. For example, we are crossing only with the recessive individual, that is with TT, with the dominant phenotype. It may be either TT. For example, if TT, that is homozygous TT, is crossed with the TT, so what will be the offspring that is produced will be, everything will be tall, right? So when you put the Punnett square, you can easily find out that this T will, this is the F1 generation that is produced, all will be of the heterozygous type. But if this heterozygous parent is crossed with the recessive parent. So here what will be the ratio that we will get? So if TT is crossed with TT, so first it is TT and here it is TT and here it is TT. So what is the ratio here? It will be, that is in the ratio of 1:1. That is 2 tall and 2 dwarf. That is 1:1 ratio if it is a heterozygous parent, right? So we can find, by means of the test cross, we can find out whether it is a homozygous tall or a heterozygous tall. Okay, clear? So that is the test cross. So crossing of an organism with dominant phenotype to a recessive individual. And what do you mean by back cross? It is a cross between a hybrid and its any parent. So with the hybrid, and if it is crossed with the any parent, then it is called a back cross. So test cross is used to find out the unknown genotype of the character, right? Example, flower color or any type of character you can find out. So back cross and the test. So here we are taking the homozygous recessive, that is WW. This is a recessive, and if it is a, we are not sure about the phenotype of this parent, right? So here it may be either WW or it may be a heterozygous. So if it is the homozygous, here all the flowers are violet. The same thing we saw as the previous slide, right? So here we took the example as the tall and dwarf. So here they took the example as the violet flower, which is the dominant character, and white, which is a recessive character. So here, if it is a homozygous type, then all the flowers are violet. So if it is a heterozygous type, half of the flowers are violet and half of the flowers are white. So then if the flowers, all the flowers are violet, then we can conclude that that flower is a homozygous dominant. And if that unknown flowers, after first the one generation, if we find violet and the white flowers, we can conclude that the flower is a heterozygous flower. Clear? So this is the basis for test cross. So based on this, actually, Mendel proposed the laws. That is, the first law is called the law of dominance. So what do you mean by law of dominance? The characters are controlled by discrete units called factors. That's what we saw till now, right? So the characters are passed from one generation to the other generation through the genes, and he called it as factors. They are controlled by discrete units called factors. And these factors occur in pairs. So they always occur in pairs. That's what that is, either it may be in TT or T, heterozygous or homozygous dwarf. So in a dissimilar pair of factors, one member of the pair dominates. That is, it is a dominant character and the other one is a recessive. So in dissimilar pair of characters in the sense, so in the heterozygous condition, among these, only one will be a dominating character. So here the capital letter T is dominant, the other one is recessive. So the law of dominance, actually, it explains the expression. So what is the role of it? It explains the expression of only one of the parental characters in the F1 generation, right? We saw that only one character is expressed in the F1 generation. So that is explained by the law of dominance, because that is the dominating character. Expression of only one of the parental character in a monohybrid cross. In the F1 generation, right? So expression of only one of the parental character. So this is an important term of the parental character in a monohybrid cross in the F1, and expression of both in the F2. And also it explains the proportion of the ratio of 3:1. That is 3:1 in the F2 generation. So how it is obtained? Because of the presence of dominant and the recessive character, right? So the characters are controlled by discrete units called factors, and they occur in pairs. In a dissimilar pair of factors, that is in the heterozygous condition, only one of the pair dominates, and that is called the dominating character, and the other one is called the recessive character. So this is called the law of dominance. And the second law is called the law of segregation. So what does this law say? That is during gamete formation. So that is when this is present in the parent, it forms gametes, right? So only one of the gamete, it fuses with the gamete of the other parent. So the parent may be either T or a small t, or it may be capital T, right? This is from the other parent. So during gamete formation, the factors of a character pair present in the parents, they segregate from each other. So these factors, they segregate from each other such that gamete receives only one of the two factors. So of the two factors in a parent, the gamete receives only one, right? So this is the law of segregation. So these factors are segregated in such a way that the gamete receives only one factor. So if the homozygous parent, they produces similar gametes. So if this is a homozygous gamete, they produces similar gametes, that is both the gametes will be of the capital T. But if it is a heterozygous parent, that is TT, produces two type of gametes. One will be of capital T, the other one will be of small t, right? They produces two kinds of gametes and each having one allele with equal proportion. So both will be of equal proportion. So it is the most fundamental principle. Actually, this law of segregation is the most fundamental principle of hereditary. So they are segregated and one of the factor is present in the gamete. So it is also called law of purity of gametes. Because each gamete that is formed is pure by itself. So either they have the dominant allele or a recessive alleles. Either dominant or a recessive allele. So clear? So this is called the law of segregation. So tomorrow we'll see in detail about the dihybrid cross, that is about the inheritance of the two characters. So today is only about the inheritance of one gene, that is only one character, either the stem height or the flower color. So based on that, these two laws, that is law of dominance and law of segregation, is more important, right? So any doubts on this? That is inheritance of one gene, and we have to be clear with the important terms here, and also about the Punnett square and how the cross occurs, like monohybrid cross, that is for a particular character. Clear? So we'll discuss few questions. An allele is dominant if it is expressed in second generation, homozygous combination, heterozygous combination, both homozygous and heterozygous condition. So which is the right answer for this? When shall we call it as a dominant allele? If it is expressed in the second generation, homozygous combination, heterozygous combination, both homozygous and heterozygous condition. Yes, D is the right answer. So the character allele is the dominant when it is expressed in homozygous. For example, if you take the tall, it should be expressed in TT and also in TT. So both homozygous and heterozygous conditions. So D is the right answer. An organism with two identical alleles is said to be hybrid, homozygous, heterozygous, and dominant. When two identical alleles. Yes, B is the right answer. So a test cross enables one to determine the viability of the cross, distinguish between homozygous dominant and heterozygous dominant, determine whether two species can interbreed, determine the similarities in the DNA of two species. So which is the right answer? A test cross enables one to, yeah, again B is the right answer. So it enables one to distinguish between the homozygous dominant or the heterozygous dominant, right? A cross between hybrid and the recessive parent is hybrid, and the recessive parent is back cross, test cross, monohybrid cross, and dihybrid cross. Yes, B is the right answer. That is, the hybrid is, for example, TT with recessive parent. So this is called the test cross. So the different forms of a gene are called heterozygotes, alleles, supplementary genes, and complementary genes. So the different forms of a gene are called, they are called alleles. That is T and T. So these are the different forms of the gene and they are called the alleles. So which one of the following is the genotypic ratio in the monohybrid cross? So if you take a monohybrid cross, that is only one character. So which is the genotypic ratio? 9:3:3:1, or 1:2:1, 9:7, and 3:1. Yes, B is the right answer. 1:2:1 in a genotypic ratio in monohybrid cross. A test cross is carried out. So what is the purpose of test cross? To determine the genotype of a plant at F2, predict whether two traits are linked, to assess the number of alleles of a gene, to determine whether two species or varieties will breed successfully. Yes, A is the right answer. So a test cross is carried out to determine the genotype of a plant at F2. So what is the genotype? Whether it is a homozygous type or a heterozygous type. So clear, right? So maybe we'll discuss few more problem related questions. Okay, the first question is on crossing the two heterozygous tall plants, right? So we are crossing two heterozygous tall plants. A total of 500 plants were obtained in F1 generation. When these two were crossed, the F1 generation, that is a total of 500 plants were obtained. So what will be the respective number of tall and the dwarf plants? Tall and dwarf. Right, the options are 375 and 125, and option B. So this is option A. Option B is 250, and option C is 475 and 25, and option D, it is 350 and 150. So what is the correct answer? That is, the question is, on crossing two heterozygous tall plants, a total 500 plants were obtained in the F1 generation. So what will be the respective number of tall and dwarf plants obtained in the F1 generation? So what will be the answer? Maybe you can write it in the form of Punnett square, right? Yeah, A is the right answer. So how do you find out? T and T, and so here you are crossing the T and T. So here it will be TT, T, and here it is TT. So here in the F1 generation, the plants that are obtained will be 3:1, right? So 3 tall and 1 dwarf. So the ratio is 375:125. Okay, clear? So the next question. In mice, black coat color, that is black coat color, which is represented as allele B, is dominant to brown. Coat color, which is represented as B, okay, that is small b. The offspring of a cross between the black mouse and a brown mouse were allowed to interbreed. So listen to the question, that is, offspring of a cross between a black mouse and a brown mouse were allowed to interbreed. So what percentage of progeny would have the black coats? So the options are 25%, 50%, 75%, and 100%. So the question is, yeah, it is option C. So how did you find out? First thing, we have to find out the first step, that is black coat color is dominant to brown coat color. So the offspring of the cross will be BB, right? So the offspring of a cross between a black mouse and a brown mouse were allowed to interbreed. So this is what will happen. So it will be 75%. Okay, clear? So we have to be clear with the ratio. So that is important in this. Then the next question is, on which plant Mendel had carried out his investigation, whether it is a garden pea, wild pea, cow pea, or pigeon pea. Yes, it is A. During breeding, the removal of anthers from a flower is called anthesis, pollination, emasculation, and vasectomy. Yes, emasculation. That is the removal of anthers. Mendel formulated the law of purity of gametes on the basis of dihybrid cross, monohybrid cross, back cross, and test cross. He formulated the law of purity of gametes on the basis of dihybrid cross, monohybrid cross, back cross, or the test cross. So law of purity of gametes is nothing but the law of segregation, right? So no law of segregation. So law of segregation is based on the monohybrid cross. So B is the right answer. So he formulated the law of purity of gametes on the basis of monohybrid cross. That is law of dominance, and law of segregation is mainly based on the monohybrid cross. Then in a monohybrid cross, what is the ratio of homozygous dominant and homozygous recessive individual in F2 generation? That is whether it is 1:2:1 or 2:1, or it is 3:1 or 1:1. In a monohybrid cross, what is the ratio of homozygous dominant and homozygous recessive individuals in F2 generation? Yeah, D is the right answer. That is 1:1 is the right answer. So the cross between a recessive to its hybrid or its F1 plant is called. The cross between the recessive to its hybrids or its F1 plant is called back cross, test cross, monohybrid cross, and dihybrid cross. Yes, it is a test cross. Then what is the genotypic and phenotypic ratio of monohybrid test cross? Genotypic and phenotypic ratio of monohybrid test. It is 1:1, 1:2, 3:1, 1:2:1 of a test cross. So the test cross ratio is 1:1. That is, it is a genotypic and phenotypic ratio of a monohybrid test cross is 1:1. Then, so in Mendel's experiment, the color of seed coat, nature of flower, position of the flower, color of the pod, and height of stem are called alleles, genotype, phenotype, and all of the above. So all these are called alleles, genotype, phenotype, and all of the above. Yes, see, that is phenotype. All these are the physical characters. That is color of the seed coat, nature of the flower, position of the flower, color of the pod, and height of the stem. All these are the physical characters. That is phenotype. If 120 plants are produced on crossing pure red and pure white flower pea plants, then the ratio of offsprings will be 90 red is to 30 white, 30 red is to 90 white, 60 red is to 60 white, and all red. If 120 plants are produced on crossing pure red and pure white, pure red and pure white. So the crossing between a pure red and a pure white pea plants, then the ratio of offspring will be, what will be the ratio of offspring? Total plants are 120. So 90 red is to 30 white, or 30 red is to 90 white, or 60 red is to 60 white, and all red. So what would be the answer? So here, how, for example, if pure red, yeah, D is the answer, because everything will be red, all are red. D is the right answer, because that is the dominant character. The Mendel's law of segregation is based on separation of alleles during Gamete formation, seed formation, pollination, embryonic development. Mendel's law of segregation is based on the separation of alleles during gamete formation, seed formation, pollination, and embryonic development. Yes, A, that is gamete formation. And in a cross, 45 tall and 14 dwarf plants were obtained. The genotype of the parents was, that is, the result is 45 tall and 14 dwarf. So the options are T, T. This is option A. And option B is homozygous tall, which is crossed with heterozygous tall. And option C is heterozygous, that is both are heterozygous. And option D is homozygous tall with homozygous recessive. So which is the right option? In a cross, 45 tall and 14 dwarf plants were obtained. So these are the resultant offspring. So the genotype of the parents was, so what is the genotype here? Tall and also dwarf was present. So what will be the thing? So you analyze, if you take the first thing, all will be tall. If there is a cross between these two, there will be no formation of dwarf, right? So if you take this, here also what will happen? Only either homozygous tall or the heterozygous tall will happen. There will be no formation of the dwarf. They cannot be produced. But if you take this D, what will happen? Here also, you write and see. One combination, everything will be tall. So which will be the right? You are getting the dwarf plants also. Yes. Now you are able to get it right. So D is not the right. So the correct option is C. So you have to analyze. So there they gave the parental and we analyzed the offspring that is found here. So here they gave the offspring or the progeny that are produced, and you have to find out the parent, right? So based on that, based on this factor, you have to analyze what will be the genotype of the parent. So C is the right answer. So are you clear? So are you able to follow these calculations, right? So any doubts in this? Okay, so the next question is, which technique is used by Mendel for hybridization? Emasculation, bagging, protoplast fusion, 1 and 2 both. Which technique is used by Mendel for hybridization? Emasculation, bagging, protoplast fusion, 1 and 2 both. Yeah, D is the right option. That is, he used both emasculation and bagging. So when a plant have two alleles of contrasting characters, it is called homozygous, dioecious, heterozygous, monoecious. When a plant have two alleles of contrasting characters, it is called homozygous, dioecious, heterozygous, and monoecious. Yes, C, heterozygous is the right answer. Then, heterozygous tall plants were crossed with dwarf plants. What will be the ratio of dwarf plants in the progeny? The options are 50%, 25%, 75%, and 100%. Heterozygous tall. Heterozygous tall plants were crossed with dwarf plants. What will be the ratio of dwarf plants in the progeny? Whether it is 50%, 25%, 75%, or 100%. Yeah, this cross. So the 50, A is the right answer. That is 50% will be of dwarf plants. Okay. A pure tall plant can be differentiated from a hybrid tall plant by measuring length of the plant, by spraying gibberellins. If all plants are tall after self-pollination, if all plants are dwarf after self-pollination. So how will you find out a pure tall plant from a hybrid tall plant? So by measuring the length of the plant, or option B is by spraying gibberellins. Option C is if all the plants are tall after self-pollination, or option D is of all the plants are dwarf after self-pollination. Yeah, C is the right answer. So if all the plants are tall after self-pollination, then we can find out that it is a pure tall plant. Okay. Allele is the option A, alternative form of a gene pair. Option B, total number of genes for a trait. Option 3, C, that is total number of chromosomes of a haploid set. And option D, total number of genes present on a chromosome. Allele. So what is an allele? Allele is the alternative form of a gene pair, or total number of genes for a trait, or total number of chromosomes of a haploid set, or a total number of genes present on a chromosome. Yes, it is option A. That is the alternative form of a gene pair. Right? Genes do not occur in pairs in zygote, somatic cell, embryo, gametes. Genes do not occur in pairs in zygote, somatic cell, embryo, and gametes. Yeah, D is the right answer. That is gamete. One of the following did not constitute the 7 contrasting pairs of characters noticed by Mendel: height of the plant, shape of the leaves, shape of the pod, and color of the pod. Yes, B, that is shape of the leaves. So he didn't take into account the shape of the leaves. So according to Mendelism, which character is showing dominance? Terminal position of a flower, green color in seed coat, wrinkle seeds, green pod color. So which is the right option? According to Mendelism, which character is showing dominance? Terminal position of the flower, green color in seed coat, wrinkle seeds, and green pod color. Yeah, D is the right answer. So you have to be clear with these, which one is dominant and which one is recessive. Okay. Then, yeah. So I hope you are clear with this calculations till now, right? About the monohybrid cross. Yeah. Okay, tomorrow we'll see about the dihybrid cross and how to draw the Punnett square for a dihybrid cross, and about few calculations based on the dihybrid cross. Okay. So as far as this inheritance of one gene is concerned, you be clear with the different terms, and also about the back cross, test cross, and about the ratio that is obtained in the monohybrid cross, and based on this few problems they may ask. Okay. So you go through it and you practice some questions also regarding this. Okay, so then thank you. We'll continue tomorrow, right?